So I really appreciate your time, Dara, that you're spending time with us to learn about the microbiology labs and see how it all works. And I hope that I can teach you a thing or two on how to do microbiology labs online. So it should be pretty exciting. And let's just get started. No wasting time here.
Sounds great.
Okay. So all the activities were designed by myself, and it's basically based on our microbiology lab curriculum, what we usually do at the University of Florida. And we try to do the same learning experience in the online environment as well as in the lab. And so in order to do some of the replication of the hands-on activity, we have the Carolina home kits, which also can be used, ordered from other organizations, but you already have the kit. And so we're using the Carolina kits with all the activities that we can learn microbiology hands-on skills. All right, so the first, what I want you to do, you open your kit already, I assume, and I want you to go and get your PPE, your personal protective equipment. And that's in the bag that contains the goggles, the lab coat, and also your gloves.
Okay, I'll get there right now.
Perfect.
Okay, so I have this bag, and inside, I'm assuming it's this kit, right?
Exactly right. That's your
Okay.
personal protective equipment.
Perfect. Okay, yes, I see goggles, gloves.
Right. So if you can unpack the apron, and you have to attach the strings on the apron so you can tie it around your neck and your back. It's just like a shoelace. It's pretty easy to tie on.
Okay, here we have the apron.
And you see the shoelaces, you just have to tie it on the top and on the sides.
Yes. Which side? I'm guessing that this goes around the neck, this part, and then.
Yep, that's right.
Alrighty.
But did you see the shoelaces?
Ah, yes, they fell out. Yes.
Yeah, so you need to tie it on, otherwise it doesn't get to stick.
Okay. Okay.
So tie one on the neck part and one on the side part.
Alrighty. Let me see.
And just on a side note, all of the activities are approved by Environmental Health and Safety at UF. So it's all biosafety level 1, and you should be able to do the experiments without any problems. Even if you have underlying conditions, pets, or young children in the household, there should be no problems.
Perfect. Just to make sure, does the shoelace go through the loops like that, and then I just tie it around the back?
I would make knots on each side.
Okay.
So make a knot on each side, and then you can just loop it around your back.
Okay. Sounds good. Okay. Knot here. Okay. You know, I think I did this incorrectly because I think it goes like this, yes? That's the orientation, right? To have it.
Yes. The thinner part is on top.
Yes, okay. So I did a knot around this one and this one, but I'm guessing that's incorrect. So it would go between these two, correct?
Yes, yes.
Okay.
That's it.
So I will untie that. It takes a little more creativity than those those robes that we had in lab.
Yes,
Those are just
definitely.
a simple shoelace knot. Okay. Okay, so I will take it through here. Okay, so once I have this, just tie a knot on each side, and that way it doesn't go anywhere, correct? Just like that?
Yep.
Okay. Just here. Okay, almost done. Just doing the bottom part now.
Okay, perfect.
And voila. I think I'm done.
Perfect. Excellent. And then see if you can grab a pair of gloves and put on the gloves as well. And we can spare the goggles. They're
Okay.
optional for biosafety level 1 because
Perfect.
we are
Showing the glove bag. Okay.
perfect. Excellent. Here we go. We're ready to go. So we've got our PPE. What does PPE stand for again?
Personal protective equipment.
Excellent. And again, the goggles are optional in biosafety 1. So we're ready to go. The biosafety levels are really important to understand because it depends on how you have to protect yourself and your environment. What we do is biosafety level 1, which is very, very low risk microbes. And so there should be absolutely no danger to you or your environment. Aseptic technique, that's sort of the bread and butter of microbiology. An aseptic technique is like a method to prevent any kind of spoilage or contamination. And it's not just to prevent contamination of your culture, but it's really protecting yourself, your environment, and your culture from being contaminated. And that's one thing we will do throughout every single experiment. So aseptic technique is the bread and butter of microbiology. All right, so the first thing what I want you to get out of your kit is the hand washing technique kit. And that's basically the glow germ and the little blue light.
Okay.
UV light.
So I see this bag that says hand washing technique.
Perfect. That's exactly what it is.
Perfect. And the only thing I don't see is the glow germ light. Is that what's wrapped in here?
It should be just like a little black device.
Oh yes, I think it's wrapped up inside the paper here.
It's just like, yeah, it's just like a little light. It just looks like a tiny flashlight.
Yes, it's in this bag, in this glow germ bag.
Excellent. All right, and what we do right now is not the whole exercise, but I just want you to see how you can actually clean your surface, your working surface. And assume you have either some ethanol or diluted bleach. Do you have that already, or do you want to go through the steps of how to dilute it?
I don't. So I don't have ethanol or diluted bleach. Would I be able to? Let me actually ask. My mom might have some bleach. Let me ask her.
Okay, sounds good.
Okay, I'll be right back.
Sure, sounds good.
I have my mix, 9 part water, 1 part bleach.
Excellent. So what we're gonna do, which is what I want you to do is take the little glow germ, and you have a working desk in front of you where you can work on, right? A surface that's safe to work on.
Yes, it's just my desk. Would I need some kind of board some kind of board to do experiments on?
Not really, no. As long as and bleach should be fine. It shouldn't damage anything as well. So take your glow germ and take a little dab and put it onto your desk. And if you can show us with your camera, if that's possible.
Yes, I'll just take my glow germ out and I'll point the camera down.
Yeah.
Let's see. Okay. 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 So I'm gonna try to point it down as much as possible. Let me see. Is that all right? You can see my desk?
Yep, that's perfect. That's excellent.
Okay.
So put a little bit of glow germ on it, just a drop, and rub it around with your hands.
Okay, so I'm just opening it and just squeezing it a bit.
Yep. Perfect. And then just use your glove and rub it around a little bit.
Okay.
All right. And now if you can take the light.
Let's see, there we go. Okay.
And shine the light on it. Maybe, I don't know if you need to turn off the room light.
Okay, yes. I can shut that off.
And then we see the mess you've made. Perfect.
Okay.
Okay. You have to take out the little tab so that the battery gets activated. Perfect.
Yes, okay. Okay.
Now it should work. Here we go. So you can see the shining UV light that illuminates the glow germ. Perfect. All right, so you made a mess, and it could be as much as a COVID contamination. It could be any other bacterial pathogen contamination, and it's now up to you to clean it up. So use your diluted bleach and some tissues.
Okay.
And see if you can clean up your desk.
Okay, I'll get a piece of paper towel to clean this up.
Perfect.
Okay. I'll just dip this into.
Why don't you just wipe it off with a paper towel only?
Okay.
And then see if it comes off.
Okay. Let me do that right here. And would you like me to illuminate it with the blue light so we can watch?
Yep. See if you can clean it up. It's pretty clean, actually, right?
It is pretty clean, but
yep.
I still see some. If you look you can still see the shadow here, but it's pretty clean.
All right, let's maybe try to get a fresh tissue and use some bleach and hopefully make it even cleaner.
All right. Okay.
All right. Perfect.
Now it looks completely gone.
All right. Now let's look at your glove, because you spread it with your glove. See if your glove is still contaminated.
Yes. So
What do you think?
I think I see a little on my pointer finger.
I see quite a bit from what I can see. So it's
Yes.
still, the gloves are still contaminated. So it's really hard to detect contamination because it's invisible.
Right.
And that's exactly what happens with the COVID spread as well, because it can be on surfaces, it can be on people's hands, it can be on your hand. You infect yourself by touching your face. So that's the tricky part about cleaning, with aseptic technique.
Right.
Make everything really clean.
Right.
I think we have a good start. All right. So if you want to, you can always take your brother or your parents or sister or other relatives, give them a little bit, rub it in their hand, and send them to go and wash their hands and see how good they do.
Right. I remember we did the experiment where we washed our hands and we would test how well we washed our hands. And
Yes.
those that didn't scrub between the fingers, on the palms, everywhere, we'd still have glow germ all over.
That's right. Yep, that's right. Especially for everyday people, and especially professionals like doctors and dentists, it's really important to clean.
Absolutely.
At all times. All right, perfect.
All right.
So I'm gonna continue share my screen. So we did the first activity. This is great. Perfect. And basically we just used the low germ UV light and then looked for contamination either on the surfaces or on your hands. And one of my students made a video about the hand washing, which is a pretty good demonstration that just wiping it with tissue usually doesn't work, but you have to really scrub it properly. And again, especially right now. All right, so we did that already. And this is our sign for when you go into action. And these are actually two of our graduate students, Maddie and Artemis. And basically just a sign for you get to work, but you've done this already. So let's move on to the next section. So the next part, what is really important is we need to have cultures we can work with, because we do streak for isolation, we do enumeration and some other activities. So we need active live cultures to grow. I want you to go back to your Carolina kit and find the microquick cultures. You should have two different vials. And I want you to use the Staphylococcus epidermidis. And it's a little vial with a dried pellet. And I want you to have a liquid soy broth, which we inoculate. And then also get one of the tryptic soy agar slants, which is a solid agar. And we will create our cultures.
Okay. Perfect. 1 second. Okay. So I have tryptic soy broth.
Perfect.
I have tryptic soy agar.
Yes.
Which is much more viscous and less liquid-like.
Right.
Okay, so I'll put those off here. And I created this little box that holds the tubes from the kit.
You should be also able to have a rack. So your kit should come with a little tube rack.
Yes, I believe I saw that. But putting this together, I just slapped this together really quickly, and it seems to be holding it up together.
That works as well, but for other experiments, we'll use that later on.
Perfect. And I will put these here. Okay, so I have my two of these, and now the Staphylococcus epidermis.
Perfect. And then we also need a Sharpie or the wax pencil.
Yes. Okay, let me see if
and we also need a transfer pipette, a sterile transfer pipette.
Yes. Okay. I have, so this is where we get the sterile pipettes?
That's one of them. There's another whole bag with pipettes as well, a larger bag.
Yes, I see. Pipettes. I see. I have it right here. Pipettes here.
Perfect. Yes. Sterile transfer pipettes.
Perfect. Okay.
And you've got a pen?
Yes, I have a Sharpie right here.
Excellent. So the key thing when you work in microbiology labs, you have to label absolutely everything. And what we usually label it with is your name or initials, the date, the media you inoculate in, and the organism or the sample you use to inoculate the media.
Yes.
So it's always initials, date, media, culture, or origin. Okay, so if you label your broth and your agar slant.
Okay, I'm currently labeling the broth because I didn't do that, the E. coli broth that I did the last time, but let me label these, the ones we're doing right now with the Staphylococcus. So I would label it 12/23, today's the 23rd of December as we're recording this. 12/23. And then my initials, so DL. And then you also mentioned the sample that we're using. So I would write, what's a shorthand way of writing that we're using this Staphylococcus?
SE, Staph
SE.
epidermidis, SE is fine.
Okay, perfect.
And because the tube is labeled already with what media it is, you don't need to write the media, but sometimes you get plates that are not labeled. And so we need to write the media on the label on the tubes or on the plate as well.
Okay. Perfect.
Okay.
So would I label the agar as well or just the broth for now?
Both.
Both? Okay.
Yeah.
I'll label that as well. Okay.
All right, and then we need two more things. You need your little flame, the paraffin light, because we want to practice aseptic technique.
And
Some of our students who live in dorms can't use it because they're not allowed to have flames, even if it's for scientific use.
yes, what I also use, and I know some people might have this, is this little lighter thing, which might be helpful.
That absolutely works, yes.
But in which bag would that Bunsen burner be found in?
That would be in the large bag with sort of everything, with the PPE and the pipettes and everything else. It's usually in here. And you also can see the rack, the tube rack in here.
Yes, okay.
It's a little paraffin candle with a wick. If you take the lid off, it's just like a little light. Yeah, and so this, if you use this container, Dariel, and take one of the empty plastic bags and put it in here, that's a nice container to put your contaminated loops and your contaminated cultures if you don't need it anymore.
Perfect.
So I always have a container on my workstation to have contaminated materials to put it in there.
Perfect. Okay, and I just found the Bunsen burner. It's, in case anyone was wondering, it's wrapped in the brown paper. Okay.
Perfect. So you take the lid off, and then sometimes you may want to tip it over a little bit just so that the wick gets wet. So tilt it over so that the wick gets wet, and then it lights a little bit more easily.
Okay. Okay, I just want to put a bag, like you said, over this container. And you said the bag's from this, from the PPE kit or from another?
Any empty plastic bag will do, just to put it in there so that you can just take it out and then dispose of it.
Perfect. Okay. I don't think we have any plastic bags here. Let me see. I'll just put this one here. Okay.
So
Okay.
any empty plastic bag from the kit, you can just put in here and then sort of
Perfect.
slide it over.
Okay, so tilt it over to get the wick a little wet.
Mm-hmm.
And then just light it with the fire device.
Uh-huh. You want to show it to us in front of the camera?
This thing sometimes has problems working, but let's see if I can get it to light. There we go.
Perfect. Sometimes takes a little bit.
There we go.
Great. Excellent. So we can have proper aseptic technique, almost like in the lab. Great.
Okay, so I'll put this down and let me put the camera in the same position I had before so we can see my desk.
Excellent, yeah.
Point it down. There we go.
And so the key thing, the key part, that looks great. So the key thing for microbiologists, whenever you open any tube or any bottle or anything, you always quickly hold it over the flame so that the mouth of the container gets sterilized and you don't contaminate your culture. So if you take now your dehydrated culture, if you take your staph epidermidis, and you can take off the little metal cap. There's a little perforation. So you may have, I don't know if you can get in with your fingernails or like a little tweezers or something, but you have to pull it off. Yeah.
So I use the little pin I have here. Let me see if I can.
There's some tweezers or pin or something just to pull it up a little bit, to loosen it. All right, and then take off the metal rim.
I had another pin here that was a little thinner. And this part was a little tricky at first to get off, but you're trying to get off this little aluminum.
And don't rip it off, just pull it off so you can actually open the whole rim around the plastic lid.
And mine just fell off, but I'll figure out a way to get it all off.
That's perfect.
Okay, there we go. So
excellent.
The rim fell off.
And can you show the little pellet at the bottom?
Yes. I'm not sure if it's in focus. Let's see if I can get it in focus.
This is exactly. So the bottom part is like dehydrated culture. That can be, if you add media, the bacteria will start to grow again. Perfect. So we're gonna just, once you have your liquid medium ready, get your transfer pipette.
The broth, correct? Okay.
And your transfer pipette. One tip when you use your transfer pipette, never open it at the bottom because you want to keep it sterile.
There you go. Just lost the flame.
So you always open it on the top part where the bulb is, and you don't want to touch anything below.
Yes. Okay.
And you pull it out and then it's sterile. But you want to at the same time keep it like this and then have your liquid broth and flame it.
Okay. Okay, so I opened the top part.
And then take your liquid broth. And then put the mouth over the flame. Perfect. Twist it a little. Yep, perfect. And so that's sterilizing the top. And now you can take your transfer pipette.
Okay.
And take about 1 milliliter out.
Okay, so we just squeeze the tip and...
Yep, squeeze the bulb and then take about 1 milliliter.
Okay.
And then pull it out and see if you can put the lid back on your liquid broth. Can you hold it a bit further down?
Like this?
Yeah, and ideally you flame it again before you put the lid on it next time.
Okay.
Yeah.
Sounds good.
Okay, and now you use your dehydrated culture.
Let me angle this camera up just a bit so we can see more.
Yeah, that's great. Okay. And then take the plastic lid off.
Okay.
And then put your culture in here, in your liquid media in here.
Okay, just squeezing the pipette.
Yeah. And then flame the top of the tube again. So every time you open or close anything, you always flame it before and after. Perfect. And then put the lid back on. In an ideal world, you don't put the lid down, but it should be okay.
Okay.
Okay, and then swirl it around a little bit. And we'll let it sit for about 15 minutes.
Okay. And
So
just
you can dispose of your transfer pipette. And then you take your liquid slant. Now 15 minutes have passed. Go fast forward. And you take the slant, the liquid broth again. And take a new transfer pipette.
Okay.
And then open it up again. And take everything from your dehydrated culture and transfer it into the liquid broth. So take everything, not from this tube, from
this.
the other one. Yeah.
Okay. So
because you want your culture to grow.
Yes. Okay. So I will open this one. Will we flame it again when we open it?
Yep. Always flame. It's always good practice.
Okay. So. Okay. I'm putting this right side up. Okay.
Just take everything out.
Okay.
And then transfer it into your liquid broth. And flame it before you transfer it. Flame it.
Yes. Yes.
Excellent.
Okay. We're transferring everything here.
Yeah. Perfect.
Okay.
Flame
And is it
it one more
practice
time?
to flame it again after the transfer?
Always flame it before and after every transfer. And then we just completely successfully done an aseptic transfer of a culture, rehydration and aseptic transfer. Great. So shake it a little bit up and down, and then we incubate it.
Okay, I will dispose of this pipette that we used.
Great.
And I will put this to incubate.
Yep. And we have some time pass. And if you have, do you have your slant as well and a loop?
Where would I find that? If you could please show me.
So the loops are a pack of this.
Okay.
And you take a loop to transfer it onto the slant. And the slant is the agar that is sideways.
Okay, so my loops are the blue ones. Is that the
That's
same?
fine. Blue or yellow, either one is fine.
Okay. And then you mentioned the slant. Where is that to be found?
They should be in the same box or the same container where they had the media. And I think you showed it to me before. It's just the agar that's sort of like a little bit tilted.
Yes. Okay, I have that.
You labeled it already.
Yes. Here's our pack of loops.
And again, for the loops, you see this is the top part, and this is the part where you touch the culture. So always just open it on top.
Right.
And take one out carefully.
So we open from this butt and not the round loop side.
Exactly, yes.
Okay, so I'll open it there. And careful not to contaminate anything.
Yep. And you can't flame the loops because they're plastic, so you don't want to melt them.
Right. Okay, so I have my loops ready, and then would I take my slant?
So take the loop and take a little bit out of the culture. You can use that culture.
Oh, this the same?
Yeah, there should be enough left.
Okay, let's do that.
You can also use it from the other culture you just inoculated. Either one is fine.
Okay, let's do this one since we have it right here. So,
perfect.
So we take one of the
and don't forget to flame it.
Flame, yep. Okay.
You need a few more hands, right?
Absolutely. Trying to be almost lost my flame. Trying to be aseptic is difficult,
That's
but
awesome.
okay. So I just rub it around.
Check if you have enough culture, but there should be enough in it.
Yes, I think there is enough. There's some I see in the corner in the bottom.
You only need about 10 microliters for the loop, so it's a small amount.
Perfect. Okay. So I have that.
Okay.
And now would I just open and flame this?
Uh-huh.
And let me know at any time if I'm out of view of the camera because sometimes I might not notice.
No, it looks great.
Okay.
And then for the slant, just go like a little ziggy-zaggy line. Start at the bottom and then zigzag up.
Okay.
Perfect. All right, you just mastered another technique. We inoculated an agar slant. So you can put your loop, flame it again, put your loop in a container. Perfect. And then we incubate at room temperature. Usually in the lab, we would incubate it at 37 degrees, which is body temperature, the same as about 97 degrees Fahrenheit and 37 degrees Celsius. But because we're at home, so we just use room temperature and the organism should grow just fine. So just put it in your rack and then we'll come back in 2 days and look at it later.
That's the one we originally did, correct? That
Yep.
First one? Okay. And that was E. coli that we did. Okay.
And if you shake it a little bit, it should be really cloudy. The slant should, can you see growth on the slant?
Yes, okay. So I think we did it on the Staphylococcus.
Yes.
Okay, there we go. Okay. So that's the clouding one.
Yeah, definitely. Very good.
And now on the slant, we can see some growth.
Yep. Yep, definitely grown as well. Very good.
So yes.
Yeah, everything worked out great.
Perfect.
Excellent. So you can dispose of your vial. You can put the cap back on.
Yes.
Put
Would I dispose of this in my waste bin?
In your waste bin, absolutely. And then at the end, we can decontaminate everything with the bleach.
Perfect.
All right, awesome. You can also keep it for later as well.
Yes.
All right. Great job, Dariel. Okay, let's see what's next. So I'll share my screen again. And
should I just keep my camera on the desk like this?
yeah, for now, that's fine.
Perfect.
All right, so we did the rehydration of the cultures and we inoculated the agar slant. And that's what we need. I have a demo as well, but I think it's much easier for the students to understand when you actually do it. We incubate it at room temperature. And then within a couple of days, you should see that instead of it being really cloudy, you will have a really sort of, instead of being clear, you have a cloudy tube, and that indicates growth of bacteria in your medium. All right, excellent. And we already did it. Great job. And then the next part is to do the art of microscopy. So we're going to grab the microscope.
Okay. And I purposely did not set up would I put out my flame, by the way?
Yes. Yeah, please turn off the flame.
Okay. And I did not set up my microscope, so we can go over the setup process for
excellent. Excellent.
And we will go through the setup process together so that in case anybody gets stuck anywhere, you can just watch this and we will figure it out as we go. So I'm just opening the box.
That's great.
Okay. It comes with an instruction manual, but I know scientists never read those. We just go through trial and error. Okay. Okay. Let me get the boxes out of the way.
So you need to get one more thing from the box because your eyepiece is in the box as well.
Yes, I see that in this larger container.
Yeah.
Okay. Is that the white little rectangular box?
No, that has some other slides and stuff in it. So the eyepiece should be inside the cardboard box.
Okay.
And it's like a tube, like a black thing. Yes, perfect. Because what's in the microscope right now is just a plastic holder. And so you need the eyepiece to actually see something.
Okay. Okay, yes, and I see we have some other tools in this white box, but I think that's for later.
Quite some interesting things in there.
Okay. Here is our microscope.
Perfect.
Perfect. Okay. So we would attach our eyepiece, yes?
Mm-hmm.
Okay.
Perfect.
We would take off this little cap, yeah? Okay. And this orientation, yes?
Yep, that's it.
Okay. Okay. Let me see if I see something. Nothing yet, but I see it's a microscope and it's looks like it's working.
Yep.
So
All right,
needs
so
batteries.
the
Yes?
key thing is batteries, absolutely.
Okay.
You may need a little screwdriver or
Yes.
coin or something.
Let me see if this will work. It does. Okay. And I have two AA batteries. I will place.
Right.
Oh, actually, we need three.
You need three?
I'll bring one more. I had two here, but one, two. Okay. Okay, I'll put the screw back in. Okay, we are
Perfect. Good. All right, and you can see the little switch on the side.
Yes.
There's one, if you tilt it one way, you get light from top. And that's more for specimens that are really thick, like algae or a leaf or a bug or something. And then on the other side, you get light from the top. So when you look at slides, you want to have it on position 1 so that it's like transmittent light from the bottom to the top.
Yes. Okay.
And then you have your stage clips here. That's where you can mount your slides.
Mm-hmm.
And then you have your nozzle with the objectives. You can turn it around. So the shortest objective is 4x.
Mm-hmm.
And then there's a 10x, the yellow one. And then there's one more, the blue one, which is 40x. So that's the objective magnification. And you can see that your eyepiece also has a 10x magnification. So the total magnification of, for example, the 4x times 10x is 40x. Mm-hmm. So what's the maximum magnification of your microscope?
So the largest lens is a 40x, so times 10, 400x.
That's right. So 400, and that's pretty good. You can see quite a lot of things in there. All right. Let's give it a second. Let's look at
little nifty microscope. I like it.
They are actually really pretty good. So, I mean, not quite the same what we have in the lab, but definitely better than not having a microscope at all. So let's go back here. Again, so this is your microscope. You have the eyepiece, which is 10x magnification, the eyepiece tube, the head, the nose piece with the objectives, and you can rotate the nose piece. You have the overhead illumination, the bottom illuminator, the stage clips, and then the focusing knobs on the side. So the blue knobs, that's where you can focus it and have a focused image. And then you can just see it's an inverted microscope, a compound microscope, and you can see the light path on the right. Okay, so there's lots of things you can actually examine in the microscope. There's in the box that you just looked at, there's some pre-made slides. There are some brine shrimp eggs where you can hatch them. There's yeast. You can collect things from the outside. You can make your own hay infusion, and you can really examine a lot of different things. Okay, and again, the next step is for you to look at things.
Great.
It takes a little while to actually see in the microscope. Because in the beginning, it's just confusing. It's just like playing an instrument where you don't know in the beginning, but it takes a while to actually learn and listen to your instruments. It's learn to see what's in your microscope. And I highly recommend just to try out different things. Look at the pre-made slides because it's easier to see, and then make wet mounts of things from the outside. And I really recommend you to draw, sketch, or take pictures of what you see in the microscope so you can document it for electronic lab notebook. And there's a really cool website, the virtual pond dip, which will help you to identify organisms. And we can look at that website quickly, where you can actually see, it looks like a pond dip, and you just click on different organisms. For example, these rotifers here, you click on it if you see something that looks like it, and you can learn about the organisms.
Dr. Oli, I don't believe your screen is sharing.
Okay. All right.
I remember that website was very helpful when we were trying to identify a different specimen in the lab.
Yes, so that's a good website. The link is on the PowerPoint as well. All right, see if you can maybe just take one of the prepared slides from the box, the brown box in the microscope. And there's like a little slide box with prepared slides.
This little white
That's
box?
right. Yep, that's perfectly right. And there's like a plastic container in it that has some prepared slides in it.
Okay, yes.
And it's got some wool, some frog blood, and some onion cells. Yep.
Perfect. Okay. Okay. Yes, I see a bunch of different slides.
Yeah, so there's some empty ones for your own investigation. And let's maybe start with the onion cell.
Okay. Let me see.
And then the frog blood.
Yes, okay, it's the second one in my kit. Onion bulb epidermis.
Yes. And you can do that really easily on your own as well, but let's just look at that slide. So take it and put it in the stage clip so it's safely mounted.
So would we just move the stage with our fingers, or would we adjust it with this?
No, you can just slide it underneath here. Lift up the clips a little bit and put the slide inside.
Okay, I understand.
Yeah, that's it.
Okay. And we want to have the little smear of the onion bulb right in the middle of that opening.
In the middle where the light is, yes. So if you turn on the light,
and
the specimen should be
the first or the second light setting, from the top or the bottom?
The bottom, because it's a slide.
Okay, so that's light setting number.
That's the top. Perfect. Yes. And then we start with the 4x, and then take your focusing knob on the side, because you need to make sure your specimen is in the right plane of focus.
Yes. Okay, I see it very clearly now.
right? Go maybe to the 10x, change it to the 10x magnification. And you should not need to focus any further. So it should be parafocal. It should still be almost in focus. Maybe it's just a little attachment. Okay, so what do you see?
I see little, so I see the edges kind of look like this, the edges of it around, and in the middle, it's red and purple in color, and it has, I see like streaks, lines running throughout.
Right.
It looks like long elongated cells and lots of almost like a branch-like structures.
Right, so it's basically like a lattice of cells. So the plant cells have cell walls. So they're much more rigid, but they're like almost like hexagons. And they're like many, many hexagons put together. You can also go to 40x, you probably see even more.
Okay.
And then the round thing inside is the nucleus you can see.
Just adjusting the position. Okay, let's go to 40x. And each time I change lenses, there's a little focus adjustment.
Just a little bit, yeah. But you can clearly see the nucleus, you can see the cell walls.
Yes.
That's pretty cool. And if you have a phone and use your camera and just tilt it the right way, you should be able, my former students were able to take a picture just like holding it, to the lens, and you should be able to take a picture. I've seen some actually quite impressive images. And then there's one more thing on the microscope. If you want to try it out, there's actually a dial with filters in the front.
Let me see.
And it just changes the color and the contrast a little bit. Sometimes it helps, sometimes it doesn't help.
Oh, yes. Interesting. Yeah, I'm on this red filter and it's really, and the green filter, yeah, you can really see some additional contrast. That's very cool.
Yeah, you can see different items. So we can do look at the hay infusion later on and other great things. So if you have a puddle outside or a pond, do you live near a pond?
I don't, but I probably have I remember for lab last year, I swabbed a lamppost and I got some pretty cool, like spider web sampling, and that was pretty cool.
That's definitely one thing. Another really cool thing is just take a teeth scrap from your plaque, and you can actually see cheek cells, and you also can see a biofilm on your teeth. And you, as a former future dentist, you should be able to look at, but we can do that some other time. So there's lots of cool things you can actually look at as well.
Perfect. I'm excited.
Great. So we mastered 2 more skills: the life in a drop of water and art of microscopy.
Ooh.
All right, let's look what's next.
And I'll put this onion cell away.
Right. And you can see in the box, there is the pre-made slide, but you have empty slides in the back with nothing on it.
Yes.
And then in your little container, you also have like a little box with cover slides. Because when you have a drop of water, you always want to have a cover slide on it, like a wet mount.
Yes, yes.
And we have videos to show how to make wet mounts as well, but it's important to flatten it so you actually get a good image.
Perfect.
Okay, excellent. Next chapter. Moving along.
We're on a roll.
Yep. The next part is we are going to isolate microbes. So we're growing microbes, and now we need to isolate it, because when we grow microbes, often from the environment, you see lots of different type of organisms. But oftentimes you just want to study one specific organism. So you want to have a pure culture with a single organism on it. And it's called the streak for isolation. So what you need to do, you need to get your culture, that you've grown up a couple of days ago. You need to get your lamp again. You need loops, and you need some agar plates. And then we'll perform the streak for isolation.
All right. Okay. Would it be these agar plates right here?
No, there should be with media. There should be in a box.
Okay. Just a second.
They should be in a box like this.
Okay. Let me look.
You may have put them in the fridge before.
Yes, that's correct. Okay, I'll go get those right now.
Okay.
Okay, got my box.
Perfect. That's it.
I noticed that my PPE is compromised.
All right, and grab your culture as well. The E. coli culture you made 2 days ago.
Yes. Just a second, I'll just quickly tie a knot here and we will get to work. Okay. I have my plates. Does it matter which one or?
No, they're the same ones.
Okay. Okay. So I have
So-
my E. coli. Oh, let me put it here. My E. coli broth that we mixed.
Yep, and it's already cloudy. Yep, perfect. And you have some loops, and you have your flame, and you have some agar plates and your pen.
Yes, okay. I will light my flame.
Okay, give it a second. We'll do something virtual first before we actually do it.
Okay, perfect.
It just lifts up here. And always check to make sure they're not contaminated, but they should be fine. There should be no other growth. There should be clean plates.
Yes, there's just a little bit of like condensation on the inside.
Condensation, yeah, that's fine.
Okay.
Okay.
Perfect.
All right, let's do this virtual activity. I'm gonna send you a link in the chat.
Perfect.
And then if you can open it up.
Yes.
And then you can share your screen with me and we can watch you
Perfect.
do the activity. Okay. It's a pretty neat website where you can practice streak for isolation.
Okay.
If you share your screen with,
how to share my screen. I forgot how to
It's
share
at the
screen.
bottom. It's like the green thing. Just to
Yes.
share
Okay.
screen.
I see. I see. Okay. So I'll share my screen with the animation.
Perfect.
Okay. Perfect.
That's great. And then on the left side, go to the streak plate. This is actually a really great site, which we use a couple of times for different activities.
Perfect.
And then say open module.
Okay.
You can obviously look at the instructions, but I'll walk you through it.
Perfect. Oh.
Okay, see, flash.
I don't have Adobe.
Adobe unfortunately won't be.
There we
Here
go.
we go. It's still working. Okay, so go start at the bottom. And then you take your loop and then flame the loop first. And what we do, the concept of streak for isolation is basically you're trying to dilute a culture so that you get isolated colonies, CFUs, colony forming units. So drag it into the flame.
I'm clicking and dragging. It's not letting me drag it for some reason.
It's not working?
I'm clicking on it and trying to drag.
What about clicking on the Bunsen burner first? Maybe you have to light it first. All right, maybe it's an Adobe thing.
Working.
Okay.
I see it highlights when I go over the loop, but nothing over the button.
Do you have a Mac or a PC?
A Mac.
Oh, here we go. You got it.
So I probably
need to. So hold it over the flame first. You want to sterilize it?
Is it off the Bunsen burner?
Usually over the flame.
Okay, so let me try double clicking again.
So usually you can hold it over the flame. Yeah.
There we go.
Yeah, here we go. So you're sterilizing your loop. So you're only taking specific organisms and you cool it down a little bit. And then you take one isolated colony from the plate that already has cultures on it. And you just take an isolated colony. No, that's not isolated. Here we go. Yeah, touch it gently. Here we go. And then you take it to the new plate. And then you do the first quadrant.
Okay.
Start on top and then do zigzag down.
Oh, you need to do a better job. Okay.
Oh.
So I will start right here.
It's a little bit tricky online, no doubt about it.
There we
Perfect. Go. So you can see how it's diluting out if you imagine it. And you don't want to go into the other quadrant. Perfect. All right, now you need to flame it again because you want to dilute it out. Flame it. Excellent. And then you have to touch a little bit of the previous quadrant. You don't go back. You don't go back to the first plate. You just stay on the plate where you want to do the dilution. Okay. And remember where you streaked it, so you touch it a little bit on the side.
Okay. Do we touch
on the top? I would go on the leftist side.
Like here?
Not further left. On where the red line is, a little bit further down.
Here?
Yeah. Exactly. And then start streaking, but you make sure you touch the streak a little bit what you had before.
Yes.
Yeah.
Okay.
You can go through it like.
Click the wrong.
You have to make sure you click in the quadrant to make sure you get the cultures. Go a little bit up, up.
Here?
Up, some more up. Yeah, here we go. Touch it there and then you can streak it.
Okay.
And go down and go zigzag.
Okay.
Yeah. Oh, they want you to go to the right side. Sorry.
Okay, so do I start from...
Yeah, that works.
Okay. And just zigzag from here?
Yeah. Ideally it would be the other way around, but that's okay.
Normally I know it would be from the top.
It would be the other way around, yeah. Yeah. Okay, and then flame your loop again.
Okay. Oh, it didn't, so I'll redo it.
Okay.
So would I go click here?
Hold on. Wait a second. Let me see if I can do my annotations. Wait a sec. Here we go. So what I would do, because you're diluting out this way, right?
Yes.
And this is the most dilute here. So now I would do I would take it from here and then go like this. Because you want to go from the most dilute and then dilute it further out.
Okay, understood. So I would start around here and then go like this.
Yeah.
Okay. Okay.
You need to take, yeah. Perfect, that's it. Great, and then flame it again.
All right. This is a really cool animation to learn how to.
It's a really great way to learn it.
Okay, so now we
And
go
again,
back.
don't go back in the original plate, go right on that plate, and then take it from the second part, from where it's dilute.
From around here?
Yep. So this is the area where it's most dilute. Take it from here, make sure you touch it,
Okay.
and then go to the next quadrant.
Okay, so I'll go from here and do a zigzag down this way, yes?
You can do this way.
Okay. Understood. Okay, so I'll touch.
And if you go by accident to the next quadrant, it beeps as you use one. It's pretty good. All right, flame your loop again.
Okay.
Perfect. And then one more time, and then it's time to incubate.
Okay, so we start here and then we go this way?
Yeah, that works.
Or this way?
It doesn't matter. Either one works. You want to just dilute it out.
Okay. Or actually, that's.
Make sure you get enough of the first one, because if you don't, if there's a line, doesn't mean they're necessarily bacteria.
Is there any way to redo a streak, or it just does it like it is?
You can do it 100 times until you get it.
Okay.
But it looks really good. It looks excellent.
Perfect.
Perfect. Now we incubate it.
Ooh, very cool.
A+. Very good. See, and you have isolated colonies here in the last quadrant. Very good. And then how do you label your plate again?
So I would put initials, date, and then the bacteria I used for my colonies.
And also the plate name, the agar type.
Yes, yes. So this would this be a nutrient agar?
But that's what we're using, yes, yeah.
Perfect.
But that looks great. That looks really, really, really excellent. So we're going to do the same thing now at home with your plates and your culture. This is great.
Do I stop sharing my screen?
Yep.
All righty.
And then go for it.
Okay. So let's take it out of the box. And we would be using the E. coli sample, correct? That we made?
That's right. Because it's grown up already.
Okay. So I will take a plate. One plate. And we just need one for now, yes? I put the rest.
For now, yes. You can practice it again after you see your results.
Perfect. Okay. And I will light my Bunsen burner.
Paraffin candle. Bunsen burner would be too dangerous at home. Excellent.
Yes. I remember we had Bunsen burners in the lab.
Yeah.
Okay.
All right, you've got your loop ready, and you need to label your plate before you do it.
Yes.
Do you label it at the bottom or on the top?
On the bottom. At the bottom, meaning not the side that has the agar. So this, I guess that would be the top, correct? This part that doesn't have the agar?
The part that has the agar, that's where you label it.
Okay, so we label it below the agar.
Hold on, open it up and show it to me.
Yes.
I think it fell on the wrong side. So your agar is on the wrong side. It somehow fell out. Good observation.
Oh no.
You may check on the next plate. It may be just that plate.
I might have gotten a faulty plate.
But your lid should be empty, and your bottom part should have the agar in it.
Okay.
Yes,
Okay.
that's it. So your lid is empty and your bottom has the agar. And you see, always label the bottom, because if your lid falls off, nobody knows what's in your bottom.
Right. Okay. So I'll put this
Okay.
agar away.
You can just toss that, yeah.
Okay. So I'll put initials, today's date, and then E. coli sample,
Great.
and then nutrient agar.
Excellent.
Okay. Okay, so I have my loops ready.
Uh-huh. And then again, same thing when you take your tube, flame the mouth.
Okay.
And you can, if you want to, you can draw like a grid on the back. It may be a little bit easier to see, but it's up to you.
Uh-huh.
So sometimes it helps in the beginning if you draw like a little the segments on there.
Yes, I remember that.
Just to make sure you stay in the lines. It's like draw by number kind of thing.
So I'll do that and show the camera what that looks like.
Because it's a little bit harder when you draw to actually see where you did the streaking.
Uh-huh.
Perfect, yeah, that's great.
Perfect. Okay, so I'm opening up my E. coli broth.
Great.
Okay. I'm taking a loop again from the back end so that we don't contaminate any loops. Okay. So now I would flip this around and have the agar facing up, correct?
So you can either do it this way. You can either do it that it's on the bottom and you sort of lift the lid this way and then streak. You can also do it that you hold it sort of a little bit upside down and you can streak in the air. It's probably a little bit more advanced. But if you hold it or if you have it on the plate, make sure you don't take your lid off completely. Try to hold it like this so you don't get contamination on your plate.
Yes, okay. Okay, so I will dip my loop in. Okay, flame.
Great.
And
Can you move your light a little bit so we can see your hands?
yes, move
Perfect,
this.
thanks. Yes, thanks.
Okay. So here I have the plate. Can you see it all right like that?
A little bit to the left. Yeah, that's good.
Perfect. Let me point it a little down the camera so that we can see.
Yeah, that's perfect. Awesome.
So not going to lift all the way. I'm going to do a zigzag. Okay. So after this first quadrant, I would take a new loop, correct?
Exactly, absolutely. You just toss that loop and then pick a new loop.
Okay. A lot of boxes here. Okay. I see where I went a little zigzag, so. Okay. Now we toss that loop.
Excellent.
Take another one. Uh-huh. Okay.
One more.
And one last loop. Okay.
Awesome. All right, and now we incubate it. We always do lid down because you can see sometimes there's condensation.
Yes.
And if the condensation falls on the culture, then it becomes all mushy. So we always incubate it lid down, and you can just leave it somewhere on the windowsill or something at room temperature.
Okay.
And within about 2 days, usually it takes in the incubator takes 1 day, but at home at room temperature takes about 2 days. And you should be able to see a streak for isolation. And you take a picture and then upload it on our shared files so we can all share the pictures and see. But let's see what you're expecting to see. And we'll come back later to actually look at the results.
Perfect.
But let's see what the expectations are. And I need to share my screen, right? You don't see my screen?
No, no, not yet.
Okay. Okay. So we did our activity. And then there are a couple of different things what you can see after you get your results. So if you look at just, fictive, what do you think on the left top side, what happened on that plate?
On the left top side, it looks like it got the colonies transferred from one of the quadrants to the next, so maybe some contamination.
That's what it looks like. So again, there's a couple of different ways of streaking. There's a T method and the quadrant method are the most common ones. So this particular one is streaked as a T, where you just have the first part. The first quadrant is more like the first third of the plate. So it's more like this. And then the second part is like this. So there are different ways of streaking, and there's nothing right or wrong about it. So that's how this plate was done. But so what happened, you can clearly see this looks like E. coli up here, right? The smaller colonies.
Yes.
And you have definitely isolated colonies here, here. And then what happened to these big things here? What are these? What do you think?
So it looks like contamination because they're independent from each other.
Exactly. And they look completely different than the original culture was, right?
Right.
Yeah. So that definitely was a contamination somewhere. What about the one on the top right? It's a little bit harder to see.
The larger circle. That looks like it might also be a different type of contamination because it's different from the rest.
Yeah. Again, the streak is very nice. It's very tight and so on, but there's definitely contamination. So if you don't leave the lid on it, it's very easy to get contamination. And then the same on this one as well. What about the bottom right one? How would you judge that, or what grade would you give that?
It looks like there's no, the colonies aren't there, so it looks like nothing got on it. Oh,
right,
you're talking about the bottom left?
The bottom right.
So the bottom right one looks pretty good because it looks like there's some spread overall, and I see some independent colonies, so it looks pretty good.
Right, and then the other one didn't get a lot of isolation, and the quadrants were a little bit overlapping, I think, as well.
Right.
So it's an art. It takes a couple of tries, and again, we will try that again if it didn't work out the first time. And step annotation.
It definitely takes some practice.
Yeah, it definitely does. So this is a really good one where you see the first quadrant. Again, each person has a slightly different way of streaking. And then you can see the isolated colonies here, which you should get. And then what is a CFU?
Colony forming unit.
It's a colony forming unit. And basically what it is one organism, one cell landed here. And then it multiplied and it grew to so many organisms that you can see the colony. But it's basically a clone. One organism landed there, and then it just multiplied itself. So that's why you should always start with an isolated colony, because you know it's the same organism and there's no contamination.
Right.
All right. So we've mastered another skill. We mastered the streak for isolation.
Here's my streak for isolation. It actually looks pretty decent in the first quadrant. I'm not sure if you can see or.
Yeah, I can see it a little bit. Maybe you can quickly take a picture afterwards and send it to me as well.
Yes.
Perfect.
I will.
That's good. And what about the single colony in the middle? Do you think that came from the streaking, or do you think that's a contamination?
It looks like contamination.
It's not connected to anything else?
No. So there's two little colonies here and then another colony here.